Okay, so we've talked about transcription, we've talked about translation, we've talked about a lot of steps that require enzymes to do things correctly. A lot of these, and we'll talk about more steps actually in the next chapter that occur with DNA in a cell's life. These are a lot of steps where there's tendency or there's a potential for mistakes to be made. And so we're going to talk a little bit about mutation. I'm actually going to go back to that problem we ended class with on Friday and talk about how mutations would affect that. So you'll get to do another problem. And that's kind of the big theme for today, and we'll see how much time we have left to start on the stuff for Wednesday, because that actually could take a little while. So there are several different kinds of mutation, and I'm going to talk about a few of them and the effects they have on the protein. So one type of mutation that could occur, and typically occurs when DNA is replicated, which is the process we're going to talk about in the beginning of next chapter, but it could also happen during transcription, is called a substitution mutation. So a substitution mutation is when a single base is changed, and it could lead to something like a single amino acid change. And we'll see also it could lead to no change, and that would be a silent substitution mutation, but we'll get into that. So here's the original sequence here, and you'll notice in this DNA sequence there is one base that is different. So instead of a T, this has an A. And what that changes is that in the mRNA, so this will be the mRNA, this is the DNA, the mRNA, instead of an A, you have a U. So now instead of GAG, which codes for glutamine, you get GUG, which codes for valine. This is actually the mutation that individuals that have sickle cell anemia have on one of their chromosomes. So most people with sickle cell anemia are heterozygotes, and we'll get to that when we get to genetics. So they have two chromosomes. One, they have a functioning version. One, they have this mutated form. So one single letter changing one single codon, changing one single amino acid, has changed blood from normal red blood cells to sickle cell red blood cells, which the effect of this is that the blood cells tend to distort, or they have abnormal aggregation of hemoglobin molecules, and it causes the cells to be misshapen, which makes it harder for them to carry oxygen. It also makes them more tendent to block up in capillaries, so they get problems with blood flow as well. So there's a lot of kind of problems with sickle cell anemia. So amazing that only one single mistake could do this. And this would be something that's inherited, so this is just a change in the DNA in general. But you could imagine that this could happen during transcription, in which case you wouldn't get as globally a big problem with something like sickle cell anemia, but you might get malfunctions in the protein production in one cell at one time if it's during protein synthesis. So a substitution is when you change one single base in the DNA or the RNA sequence. Okay. Here's another kind of mutation. This is an insertion or a deletion mutation. And if you remember that on Friday I talked about reading frame and how important reading frame is for setting up how you read the triplets of RNA. So here is the original DNA sequence on the top. What would happen if between this C and G there was an addition of a single T? So what happens to the protein, do you think, that's made from this? Is it changed at one single amino acid?
Changes it all.
All right, why would it change more than one amino acid?
So if you're adding one in, it shares go.
Okay, yeah, so if you look up here, GAC was the original triplet here. This has TGA, GAC. This one GAC, this is basically, they did this so that you'd see that it changes repeatedly. This was just a series of GAC, GAC, GAC over and over again. And now this reads TGA and then CGA, CGA. So it's changed everything past that point of mutation. Here's if you insert 2. Still, instead of GAC, you've got TTG and then ACG, ACG, ACG. But look what happens if you add 3. Yep, you have a change here in one amino acid, one triplet, but then you're back to GAC. So why did that happen? Okay. So when you add 3, yeah, it gets it back into the original reading frame. So basically you've just added one triplet, one codon, and you're back now to your extra one. So you've added one amino acid here. Same thing would happen if you deleted 1 or 2. 1 or 2 is going to shift the reading frame. 3 bases is not going to shift the reading frame. So adding an insertion or a deletion of 1 or 2 bases leads to a frame shift, which is a shift in the grouping of the mRNA bases. So we're going to play with some of these in the next slide. So this is back to the one that I showed you the other day. So if you haven't done this, if you weren't in class and you didn't do this on Friday, you can go ahead and try to convert this. Those of you that have, pull it up. I'm going to ask you some mutation questions as soon as we do this. So just give me a minute to write this on the board. All right, so there's the DNA. If I was to, there's a different color. So transcription. Transcription to mRNA would give me UAA, UGAAA. Did I add too many of those? Yes. ACT. I got an extra T. Okay. C A U C G A G U U U A A C G A U. Thank you. Okay, so that's my mRNA, and then if I wanted to go to my protein, translation. So again, translation to the protein. I'm going to look for my AUG. It's going to set up my reading frame here. And I have methionine, and then LYS, X. It help me out here, CAU, histine, okay. What's after that? Arginine, and then val, and then stop, right? Okay. Okay, so hopefully you all got that the other day. So what if, what if GCT to GCA? What if this T was mutated to an A? What's that going to do? So instead of CGA in my mRNA, this is going to be CGU. And what's that going to do to my protein? So instead of arginine, I'm going to have what? CGU. I'm going to have arginine. Okay, so I had a substitution mutation in the DNA. Did it have an effect on the protein? No. Okay, this would be a silent mutation. And this happens all the time. This is something that If you've learned about evolution, you learned about natural selection pressures, this is something that would not be selected against. So this is something that's free to happen over and over and over again because there's no consequence of a mutation. So basically, this T could have been really anything here, right? It could be, you could change it to an A, you could change it to a G, you could change it to a C, and it's not going to change this amino acid because CGC, CGA, CGG, CGU, those all code for arginine, right? And this often happens. If you look at a lot of these, the third base can vary. Some of them, it can be any of them. Some of it might only be two of them. There's only a few that only have one way of making that amino acid. So methionine's one of them, tryptophan's one of them. But most of the other ones have multiple ways. And you look at leucine, there's 5 ways here, or sorry, 6 ways to make it. So a substitution mutation in the third codon, the third base of a codon, is not all that likely to cause damage to the protein. It may. If you changed this C here to something else, you wouldn't have a start, and that would mess up your whole protein, right? If you changed this T to be something else, you might not have a stop, and it might allow your protein to be continued made, and it might make a completely different protein. But so that would be a substitution mutation leading to a silent mutation. So let's say, what if instead, or in addition to this, let's see, G, where is this? This GTA, this G, was mutated to an A. So here's again a substitution mutation. The CAU is now going to be what? UAU. And what is UAU instead of histine? TYR. Tyrosine, right? So there's a substitution mutation that leads to a single amino acid change. And so that can happen, especially if it's in the first or the second base. You're likely to have one amino acid change. So in the case of sickle cell anemia, that was enough to cause sickle cell anemia. But let's say, I don't know this off the top of my head, which maybe I should by now, but histine and tyrosine, what if they both happen to be polar, or they both happen to be nonpolar, or they both happen to be acidic, or they both happen to be basic? They had similar properties to them. It might not change the function of the protein. And remember, these proteins could be huge. They could be hundreds of amino acids long. If that point mutation, if that substitution mutation happened to be in the active site of an enzyme, yes, it might have huge damage being that one change. But if it's somewhere on the end of it where it, just sticks out of or maybe it's just creating the layer that sticks within the plasma membrane, it may not matter. It may just matter that it's polar or nonpolar. It may not be all that important what exact amino acid it is. So this also might not have an effect, but it might. This one definitely doesn't have an effect. This one has a small effect, could cause a big problem if it's in a vital area of the protein, but maybe not the hugest of errors. Okay, so that's again substitution mutation leading to a point mutation, a single change. Okay, how about if, so what if this A was deleted? What would that cause? So now, let's keep that A in there, but so this is going to cause this to be deleted. So what does that do? Okay, so now instead of UA, whatever it was, UAU, I'm now reading UAC. And UAC is going to be still tyrosine. All right. But now I have GUG. And what is GUG going to be? It's going to be Val. And then I have UUU, which is going to be phenylalanine. And then I have AAC. So AAC is ASN. And then I have GAU. And GAU is asparagine. And I have no stop, so if there was anything past here, it would keep going. You're probably going to run into that poly A tail eventually. And that would just start adding a whole bunch of lysines kind of on the end of it until your ribosome falls off the end of that mRNA because there's no stop codon, right? So this is a single base deletion, leads to a frame shift. So I've changed my reading frame now, and it's now mutated everything past the point of mutation. So it's caused a change, a pretty dramatic change to the protein, right? Okay, so we've had all right, let me do one more. So what if I also deleted this G? So the C is gone, and so now I've got UAG, which is what? Stop. Yeah, so that would cause, instead of here, there'd be a stop here. So that would actually truncate my protein. So you can also have that. So by shifting the reading frame, you could insert an early stop and just make a short kind of non-functional protein. So you can make a run-on protein that makes no sense, or you can make a short protein that all by deletions. Same thing could happen with insertions because you're changing the reading frame. If I had deleted a third one, it would get it back in the same reading frame, and it would at most change that one amino acid. It might change two because it might shift it just enough to change two. Okay. So based on this, where is this? Okay. So which of the following mutations is the most likely to be damaging to the protein that the DNA specifies? So a codon, remember, is 3 bases. So all right, take a second and talk with your neighbor. Discuss what these different things would do.
Like this one. Yeah, the first codon.
We're assuming these substitutions are not being stopped.
That was over here.
So I'm saying the first base of a codon, not the first codon or the last codon.
Oh.
So it's of the triplet, the 3 bases that are in a codon. The first base of the codon, the last base of a codon, the entire codon.
No, the last one you're saying is this one. Like the third letter. Yeah. In one. If you change one.
Okay,
It stays the same.
you guys done? Final answers? Okay. All right, so if I had said which was the least likely to cause damage, what would you say?
3 base deletion.
Okay, there's one that's least damaging than those. All right, remember last base of a codon, That was the first mutation I did. It was the last base of a codon, and it went from arginine to arginine. All right, the last base of a codon is more likely than any of these other ones that could cause no mutation, right? So the last base of a codon, C, is really the least likely to cause damage because most of these codons, the last base can be anything and it can be the same amino acid. So how about above that? Let's see, a codon or a 3-base substitution. What will that change? So if you, all right, let's say change one amino acid. A codon deletion, 3-base deletion, what's that going to change? All right, it's going to take away one amino acid. A substitution in the first base of a codon, what's that likely to do? All right, change one amino acid again. So these three all really have the potential of changing one amino acid. It might be less damaging to the protein to have the same number of amino acids, so probably a substitution would be less damaging than a 3-base a 3-base deletion. Yeah, so sorry, these three have the same, but this probably would be a little bit worse than these two. Why is a single base deletion the worst thing you can do to a protein? Yeah, so single base deletion leads to frame shift. So all this is a result of a single base deletion. This was the result of 2 base deletions, but it could have been a single base deletion. So you're causing a frame shift. So all this red on the board, majority of that is due to deletions. Same thing with additions. So a single or 2-base addition would also cause frame shift. So the only one here that causes a frame shift, so a change in the reading frame, is a single base deletion. The rest of them mutate at most one amino acid. They either take away one amino acid, they change one amino acid, or the substitution in the last base of a codon, that could have no change, no effect on the protein, and you might not see it, so it could be silent. Okay, so that's really the take-home message for this part, is that mutations in the DNA or mistakes in transcribing the DNA could have an effect on the protein that's made, and a insertion or deletion mistake, especially in what we'll talk about in next chapter, which is DNA replication, would cause the most damage due to a frame shift. Mutation in just the process of either transcription or translation will be a little bit less damaging than a mistake in actually making the DNA, because it will only last for the life of that mRNA, or it might only be one single protein. If you make a mistake in translation, you might have one copy of the protein that's mutated, but the next copy that you make from that mRNA will be fine. Mistake in making the mRNA, going from DNA to mRNA, will make a bunch of protein that's mutated, but as soon as that mRNA is degraded, that mutated protein will stop being made. A mistake in the DNA is going to live on for the life of that cell, really. So mistakes in making DNA are definitely worse than mistakes in making either of these other two. And we'll talk about how DNA is manufactured in the next chapter. That's kind of where we're going. Okay, so that's just a little bit on mutation and its effects. So I'm going to spend most time on this on Wednesday, but I wanted to get started on this just so I introduced this to you a little bit, and I'm going to talk about one form of regulation as we go forward here. But there's going to be kind of 2 examples we'll go into. So we've talked about protein synthesis. You now know how DNA is converted into mRNA, mRNA into a protein. We've talked in past chapters about how important proteins are. So proteins can be things like enzymes. Enzymes can control which reactions go forward or not. So if you remember the enzyme chapter, most reactions have that activation energy that they need to overcome, and an enzyme lowers that activation energy. So without an enzyme present, this reaction probably won't go forward at all. So we looked at during the enzyme chapter at one way of regulating enzyme function. We looked at feedback inhibition, where the end product would inhibit one of the enzymes, be a non-competitive inhibitor perhaps of an early enzyme, stopping that chain of events from making the enzyme. So that's one way of regulating enzyme activity. The rest of this chapter talks about other ways of regulating things like enzyme activities. And this is done through regulating the process we just talked about, regulating protein synthesis. So being able to turn on or turn off the production of the enzyme. So in this case, this is an example of a series of reactions that make tryptophan. Tryptophan is an amino acid. Tryptophan's an essential amino acid for a lot of proteins. So your body always wants a certain amount of tryptophan present in the cells at one time. So tryptophan is a series of enzymatic reactions that lead to the production of tryptophan from a precursor. And we're going to talk about how your cells can regulate tryptophan production by controlling the genes that make those enzymes. So there are 5 genes here. 2 of them make enzyme 1, 2 of them make enzyme 3, and 1 of them makes enzyme 2. And by turning on or turning off those genes, your cell can determine whether or not it makes tryptophan. So this is a pretty simple example from bacteria. Our version of this is a little different. We don't run things in the same way as bacteria. But this will give you an idea of how this regulation occurs. And then on Wednesday, I'm going to talk about a slightly more complicated mechanism, which is called the lac operon. And that's how we control the genes that, or the enzyme that breaks down lactose. So this is what the trip operon looks like. An operon is a series of genes that are all controlled by one promoter. And operons are common in prokaryotes, like bacteria. They're not something that's found in eukaryotes very often. So we do things a little bit different, and I'll talk about that kind of at the end of class on Wednesday. So the trip operon actually is one promoter that controls all of those genes that lead to the different enzymes that make tryptophan. So you can think of it as when the trip operon is turned on, RNA polymerase is going to be transcribing all of these genes. The mRNA made from all of this DNA is going to be read by ribosomes to make different portions of the enzymes. These enzymes will then function to make tryptophan. So when the trip operon is turned on, tryptophan is being produced. When the trip operon is turned off, tryptophan is being stopped from being produced. So, because you won't be producing the enzymes that are made to make tryptophan. Okay, so if you remember back to the beginning of this chapter when we talked about transcription, transcription starts at a promoter region. That's where RNA polymerase first binds, and I said this is where regulation occurs, and so now we're going to talk about what that regulation is. So this is your promoter. The terminator sequence would be somewhere down here, and that's the gene that's being transcribed. And in this case, it's a series of genes. Okay, so part of the promoter in the trip operon is something called the operator. And the operator is between the location where RNA polymerase binds and where the gene starts. So RNA polymerase binds back here on the promoter. It goes past the operator and then hits the genes. And so it starts making the actual coding gene that codes for how to make those enzymes to make tryptophan. Somewhere else, so this is doesn't make any difference where this is found. Somewhere else, there's a separate gene called TRPR that codes for a protein called a repressor. And in the case of the trip operon, that repressor is inactive when it's first made. So now let's imagine that you just ate a huge turkey dinner. It has lots and lots of tryptophan in the tissue of that turkey. And so your tryptophan levels are through the roof inside your cells. So your cells now have plenty of tryptophan. They don't want to make any more of their own because they've got plenty from your diet to make all the proteins they want for now. So they need some way to shut off tryptophan production when tryptophan is present in the cells. And how they do that is that tryptophan will actually bind to that repressor protein and turn it from an inactive protein to an active protein. So you get an active repressor protein. So this is tryptophan, this is your repressor protein. And when that repressor protein is active, it binds to the operator. So tryptophan activates the repressor now binds to the operator, and by doing so, it's now getting in the way of RNA polymerase. Okay, so transcription will stop because the repressor is active. Okay, let's say it's now the day after Thanksgiving. You've made lots of proteins from this tryptophan, and your tryptophan levels have gone down. What do you think is going to happen? Okay, so eventually this tryptophan will be used up. What's going to happen to the repressor? Yeah, repressor is going to go back to the inactive state where it will fall off the operator, and then what's going to happen to RNA polymerase? It's going to start making tryptophan, making the enzymes that will make tryptophan, right? So that's exactly what happens. Pretty straightforward, right? So tryptophan activates the repressor binds to the operator and stops RNA polymerase from transcribing the trip operon. Tryptophan levels go down, the repressor becomes inactive again, and RNA polymerase can start transcribing. So the gene is turned on when tryptophan levels are low. The gene is turned off when tryptophan levels are high. So this is what's called a repressible operon. It can be turned off. So by default, it's turned on. So when the repressor is first made, it's inactive. So it's turned on by default. It has to be turned off by the presence of tryptophan. And this makes sure that tryptophan levels always stay pretty constant. Because as soon as tryptophan is used up, it starts making more tryptophan. As soon as tryptophan levels get too high, it shuts down the production of tryptophan. Right? Okay, so repressible operon works like this. Okay, now I'm going to tell you the story of, does that make sense? Anyone have questions about this? It's pretty straightforward. One thing to know, kind of one new thing that you've heard about is the repressor and the operator. So the repressor binds to the operator, shuts things down. Okay, I'm going to tell you the story of the lac operon, but we're not going to go into it until Wednesday because it gets a little more complicated. And I'm just going to tell you the story of the Nobel Prize work from the 1960s that was done on this that started to describe how bacteria do this. So the lac operon is a similar thing to the trip operon. It produces enzymes that do something important. In this case, the enzyme, one of the enzymes that's produced by the lac operon is beta-galactosidase. And beta-galactosidase is an enzyme that takes glucose takes lactose and breaks it down into glucose and galactose. So it actually helps you break down lactose. So any of you that are lactose intolerant, this might be your problem, is that you're not actually producing beta-galactosidase in your cells. So the study that Jacob and Monod did is they grew up bacteria cells in a broth that contained both glucose and lactose, and they looked at two things, or really three things: their growth rate over time, the presence and absence of glucose and lactose, and also the presence and absence of beta-galactosidase. So probably four things then. So what they noticed is that as time progressed, at first the cells grew really rapidly, like exponentially rapidly, incredibly fast, but they were only feeding on glucose. So lactose was not broken down at all. The glucose was broken down first. At some point here, once the glucose disappeared, beta-galactosidase started being expressed and lactose started being broken down. And then up here, when it kind of leveled out, growth leveled out again, and they got mild growth then, not as fast as they first got, but slower growth then. When growth started leveling out, they noticed that the lactose was gone, and when lactose disappeared, beta-galactosidase also disappeared. So there's several things going on here. One, they only produce beta-galactosidase here when lactose is present. So they produce beta-galactosidase here, they did not produce it there. But they didn't produce beta-galactosidase as long as glucose was present. So somehow those bacteria were able to sense when glucose was in their environment and also sense when lactose was in their environment. So we're going to talk about the two mechanisms that bacteria use to control beta-galactosidase production on Wednesday. So that's kind of the little bigger story we'll get into then. So that's enough for today. All right.